Hello, everybody. Welcome to another episode of Silence Abounds Science at Home, where we will discover home lab magic. In today's episode, we're going to look at the microscopy of algae. Algae are also known as protists. Protists are a diverse group of eukaryotic microorganisms that typically do not fit into other kingdoms like plants, animals, bacteria, or fungi. They have a nucleus and other membrane-bound organelles. Proteins can be unicellular or found as colonies of cells. They may be photosynthetic or use chloroplasts to harvest sunlight for carbohydrate production. Proteins can be found in water, damp terrestrial environments, or as parasites. We're going to take a look at 4 types of algae. We're going to look at Oedogonium, Oscillatoria, my favorite, which is Volvox, and we're going to look at Spirogyra. So let's head over to the microscope and take a closer look at these beautiful organisms. Okay, so I'm at my microscope station now, and I've got some cultures that I've got of the different algae that I just discussed. So typically when I'm looking at microorganisms under a microscope, there's a couple of ways I look at them. First is under using these, what we call Petri slides. They look like little Petri dishes. And what you do is you pipette your culture into the little Petri dish, and then this just sits on the microscope stage, and you can view microorganisms freely as they float around and carry on with their everyday life. And it prevents any squashing that could potentially happen when you just use a microscope slide and cover glass. So I first like to examine microorganisms either in a Petri slide or what we call a demo slide. These are demo slides, and basically, again, you pipette your culture into the demi slide, and you have a very thin tip, which is the viewing area. So you want to get your culture, your liquid, down into this tip. And then this is a demi slide holder. And basically this sits on your microscope slide, and this slips into the slide like so. And then you view that little tip end of the demo slide under the microscope. The only disadvantages of the demo slide and the Petri slide is that you cannot go under high magnification because the objective lens will not fit over the slides. So what I do is I view them first under low magnification, which usually is plenty. And then I make a wet mount on a microscope slide and view that under higher magnification. So let's start off and let's get looking at these beautiful organisms under the microscope and learn a little bit more about the world of algae. So I'm just going to take our first specimen and put it into the Petri slide. Okay, and that just sits on the microscope stage. Okay, so we're ready. Let's take a look. Volvox is a fascinating genus of green algae that forms colonial spheres of cells, showing a key transition between unicellular and multicellular life. Each colony is a hollow sphere made of 500 to 60,000 biflagellate cells. Cells are embedded in a gelatinous matrix and connected by cytoplasmic strands. Each cell has two flagella. Flagella beat in a coordinated way to roll the colony through water. This collective motion is an early form of cellular coordination. Most cells are somatic, non-reproductive. A few large cells are gonidia reproductive. Gonidia divide to form daughter colonies inside the parent. Eventually, the parent colony ruptures to release the daughters. Odigonium is a genus of filamentous green algae belonging to the class Chlorophyceae, commonly found in freshwater environments. It forms unbranched thread-like filaments that are attached to substrates or may be free floating. Each filament is made up of cylindrical cells joined end to end. Cells are uninucleate and have a ring-like or net-like chloroplast with many pyrenoids. Odigonium reproduces by zoospores, which are motile flagellated cells. Zoospores are formed in specialized cells called zoosporingia. Odigonium plays a role in oxygen production and nutrient recycling in aquatic systems and serves as food for aquatic invertebrates. Oscillatoria is a genus of cyanobacteria, often called blue-green algae, that thrives in both freshwater and marine environments. It is a type of filamentous cyanobacteria known for its distinctive oscillatory movement, which is where its name comes from. Oscillatoria contain chlorophyll, and phycobilly proteins convert sunlight energy and produces oxygen. Oscillatoria consist of trichomes, chains of cells, within a mucilaginous sheath, can break into homogeneous short fragments for reproduction. It is named for its back and forth swaying motion at the filament tips and aids in orientation forward, light, or in escaping unfavorable conditions. Spirogyra is the genus of filamentous green algae, known for its beautiful spiral-shaped chloroplasts. They consist of long-chain filaments of identical cylindrical cells. Cells are surrounded by a mucilaginous sheath. Each cell typically has one or more helical or spiral chloroplasts, contain pyrenoids, which are centers for starch formation. Like other green algae, contains chlorophyll A and B, produces oxygen and serves as a primary producer in freshwater ecosystem. Spirogyra reproduces by fragmentation. The filament breaks into pieces and begins to grow into a new filament. In conjunction, reproduction's two filaments are lined side by side. Conjugation tubes form between adjacent cells, and contents from one cell migrate to the other, forming zygospore. Zygospore later germinate into a new filament. Wow, I hope you enjoyed taking a look at the world of algae. As you can see, they're quite beautiful organisms, and I hope you learned a little bit more about the background about each of these organisms. If you liked today's video, please hit the subscribe button. It helps show me your support. Please hit the like and share button. It all goes to helping and supporting my channel. And I truly, truly appreciate all of my subscribers. And thank you again for all your support. It means a lot. Please leave me a comment. I like to hear from you. And also shoot me an email. If you want a PDF of today's lesson or today's video, describing algae and all that, send me an email specifically requesting the PDF for the algae, microscopy on algae, and I'll be happy to send that to you for free. So until next time, keep on experimenting. Goodbye, everybody.